Stanford University.
So I should start by thanking Tom and Bertha and Malcolm for putting all of this together. It's a great opportunity, and it's already been a fun day, and it's going to be even more fun, I'm sure. So I have this very long title that between Tom and I think we came up with. And I'm going to talk about measurement science and reference materials. But you know, hopefully, I have a lot of things to say that are actually going to resonate with some of the things we've already heard today. One of the things that I've find myself doing more and more recently is talking to groups who are dealing with some of the challenges of manufacturing of stem cells, and this community is really looking for what they like to say is standards. They'd love for somebody to have figured out all the problems and they could just check off the boxes and get something good. But I find that it's a community that really has a perspective on what they need to measure and how well they need to measure it, because they're sort of, where the rubber hits the road. And it of course starts, sort of at this scale with respect to research questions. And maybe you have an animal model that you try some things out in. But one of the things that gets really tricky very early on when people start thinking about creating products and going into a manufacturing situation is that there are a lot of changes that go on in the process. A lot of people start out in flasks, and this idea that maybe they have some good science and good ideas that they think perhaps could reach the clinic, but they often don't think about what that process is going to be very early on, and it gets it gets complicated, because through the process of developing a product, you have to scale up, you have to store, you have to be able to retrieve cells that have been stored, you have to create new manufacturing processes, and somehow, all along the way, you have to be able to compare the data that you had when you were back at the research stage to the data that you can collect on your product as it goes through this scale up, scale out, expansion, storage, and bottling process. And so it gets very complicated. But one of the things that is satisfying about dealing with this community that is in this stage is that they really know how important it is to have that comparability and to have that confidence in what they know about their product, so that they can always go back to say that they know they have the product today that they had a year ago, even if they've moved manufacturing facilities and they have a completely different set of technicians and their entire manufacturing pipeline has changed. And so that confidence in how do you know that what you have today is what you had a year ago, because it's going to take many years to develop a product. That's sort of where the rubber hits the road with respect to really having a good measurement system, a good measurement process. Also, the measurements are going to change over time as you learn more about your product. So why this is so difficult at this stage is because of the vast ignorance that we all have about, regardless of how much fabulous work has been done. This question about what are the critical quality attributes, right? What do you really even, what are the things that you should be measuring in the material that you have? What is actually important that is that you can see, that you can measure, that's going to have an effect clinically? And because usually we don't understand mechanism of action, it's really hard to extrapolate what is it about this product that is the characteristics that are really essential for clinical effectiveness. And if you don't know that, you don't really know what to measure. And so, this is where it gets really complicated for people who complain about the FDA, being so difficult and stringent, is that it's really hard to develop a potency assay or a release assay if you don't really know what to measure. And so, the kinds of questions that come up a lot is, well, how many assays do I need to have? And, of course, the answer is as many assays as you need to establish confidence that you know how to evaluate your product and its clinical response. And nobody knows how many that is out of the box. It's also, from a measurement point of view, and, NIST is an unusual organization because we think about measurements perhaps a little more than other people do, and it just drives us crazy a little bit. But when you can look at something like, do I have a pointer? Is this a pointer? I don't want to hit the wrong thing. Is it? Oh, it's that. Yeah. Okay, great. Thank you. So, like, for example, this is a preparation of retinal pigment epithelial cells. So I'll just use that as an example. You can look at a preparation and you might say, the scientists or the biologists might say, that's a great preparation. That's exactly what I want these things to look like. But the person who's thinking about measurements is going to, like me, I'm going to say, well, what is it about this that they think is so good? And if we wanted to compare this preparation with another preparation, what numbers would we put on this that we could compare with the numbers of another preparation? And so how to figure out how to take what we can visually see and put numbers on them that are reliable and meaningful is really, it's an incredibly interesting challenge. One of the wonderful things about imaging, of course, is that there is so much information in here that, in principle, we can get a lot of information out that can be very meaningful, but making those correlations is really, really difficult to do. And the other thing that we like to think about is that even if you can put a number on this, can you put a value around that number for how confident you feel about that number and what that number means? In other words, you could say, this is a fabulous preparation. I know that if I implant that, it has a 90% chance of taking and being successful, right? Or maybe you don't have that much confidence. You'd like to be able to say, well, it has a 60% chance, let's go for it anyway. But how to put a number on the confidence of the number that you have is another really serious problem that we think about a lot, and that I think is really necessary for this field. And then, of course, this question about comparability. How do you know that what you can measure here is going to be what you can measure when you get down to the actual process that you're thinking about? And let me take a step back here, because I think it might be useful for this audience to really understand NIST and FDA and the differences and similarities between us, just to put in context what we're all trying to do here. So we collaborate a lot with FDA, both laboratory collaborations sometimes. We certainly collaborate in interagency groups that discuss problems like regenerative medicine. We collaborate on standards committees, standards organizations. But we are very different, and we work with the community sometimes in different ways. And having a venue like this to really communicate with one another on a scientific level is really, really helpful. NIST differs from the FDA in that we're not a regulatory agency, and so the kinds of interactions that we can have with industry are very different from FDA's interactions. We can tell industry things, and it's not really going to have a lot of impact in terms of, changing a regulatory process or a legal process necessarily. But our missions are very complementary. They can be very complementary because what we try to do is develop measurement science that could actually make FDA's job easier by providing tools that industry could use to get better measurements. And if industry can get better measurements on their process and their product and be able to demonstrate the evidence behind those better measurements, then the regulatory process becomes a lot easier. So while I'm here, what I want you guys to do is really look at what I say and what Michael says, and then tear it apart and tell us what we should be doing, okay, to be most useful. We also, we focus on measurement qualification. So for us, that means how well do you know your measurement? We also do a lot of work on, just basic science of making, of doing research on how do you measure things, right? But in the end, it's not just can you get an answer, it's how good is that answer? How well do you know that answer? How confident are you in that answer? So that's really going to be sort of the focus of what I'm going to talk about here. But, again, I really appreciate this venue, and I want you guys to really think about, as we talk, how we can be more useful with respect to lowering the barriers to the regulatory challenges. So let me just start talking about quality of measurements. I'm going to talk a lot about our research, but I want to sort of set the stage with respect to the thought process of what we mean about quality of measurements. And there are lots of different ways of articulating this, and I'm not sure that any of us have found the best way, or if there is a best way. But my colleague Mark Salet, who's out here at Stanford, likes this three-legged stool idea, and this is that every measurement should be characterized by its uncertainty, so like standard deviations, for example. That every measurement should have some element of traceability, which is like comparability, this idea of comparability, and that every measurement should have a degree of validation, so why you have confidence in that measurement in terms of bias and accuracy, et cetera. I often talk about these issues with these words because they more parallel some of the guidance documentation that FDA puts out and gives maybe a little more explanation of what these more general concepts are about. And so, these are concepts, and they're concepts that we all know, but sometimes we don't think about them in a very discrete fashion. But precision, is that whole idea of being able to reproduce in your laboratory, but then also to replicate an experiment in your laboratory, but also to have that ability of another laboratory being able to reproduce. And of course, in this field where, personnel are going to change and locations are going to change and starting materials are going to change over time in development, that idea of being able to get the same answer under different conditions can be very tricky to do. Accuracy is sort of another thing because accuracy says that not only can this laboratory get the same answer as that laboratory, but the answer is actually correct. It's actually meaningful, and so that's a little harder to do in biology. A lot of times, one of the best ways of getting at accuracy or testing accuracy is to use orthogonal methods. So if you can run a Western blot, but then you can also do some flow cytometry and you can also do some imaging and maybe something else, then you start having confidence that you are measuring that molecular species that you intended to. But accuracy is a big bugaboo in biology because we don't really necessarily know what the biological function is and what to expect. So some of these other things, it makes these other things really, really important, like robustness. And so, robustness to determine robustness would be a series of experiments that would allow you, by which you would change subtle differences in your process and procedure and in your protocol and say, is my assay result sensitive to these subtle changes, like maybe it's temperature of the laboratory, things like this that sometimes you don't even imagine should be important, and they're not the parameters that you're systematically changing, but because your assay isn't robust, when it's in somebody else's hands or in another location, you might find differences that are purely, they're not significant differences. And in this, really complex biology that we're looking at here, we know from single cell measurements, for example, that there is biological variability that's really important. You don't want to get tripped up on the difference between biological variability and trivial uncertainty in your measurement, right? So if you've got something goofy that is leading to poor robustness, you could misinterpret that as being something significant. So it's important to, to have a handle on what variables are important and what variables are not. Things like limit of detection and response function, these can be, I think, metrics by which help you compare one answer from one day to another answer. You would expect perhaps the same response function, all things being equal. And so even if the scales are different, sometimes maybe just the shape of the response gives you some information and confidence in the result. And then, of course, there's the whole issue of what are appropriate controls, benchmarks, and reference materials. And I'm going to say something about that because these are ways in which you can compare one measurement to another. If you are lucky enough to have a reference material that actually can compare this measurement to that measurement through a reference material, that's the concept of traceability. And so these are just some of the challenges, and maybe I've said them already, but these are some of the challenges that are very specific for this area that are different from traditional metrology. So, at NIST we have a 100-year history in physical measurements, but biological measurements really are going to require some very creative thinking. Reference materials, for example, are a really hard thing to think about in a lot of the biological measurements we have to do, and I'll talk about that a little bit. When we think about the reliability of the measurement itself and a lot of the work that gets done in this context, we really have to think not only about the instrument and the reagents, but we have to think about the software that we're using to analyze as well, analyze our results with. And that might be software for analyzing genomic information. It might be software for analyzing imaging. And obviously for imaging, lots of software being used. And I'll say a little bit about that. So really, that's a really sticking point that a lot of times people just expect and accept that the software that comes with the instrument, is really valid and you can just hang your hat on it. It's really complicated to eliminate bias in the measurement method because we all come into, and it's sort of like testing assumptions, which is the other bullet. We all come into the field with ideas in our heads about how things work based on what's been proved or what's been shown in the literature before. And those may or may not be valid assumptions. And, some of these things need to be questioned. I want to give you a couple of examples of where reference and benchmarking materials are actually useful and could be useful in this field. For example, one of the things that we are responsible for in our division is reference materials for Raman spectroscopy. Very common tool that's used in the pharmaceutical industry all the time to QC, for example, starting materials. The reference materials that NIST puts out allow you to take disparate spectra and put them all on the same scale so that now you can compare the measurement you got today with the measurement that you got tomorrow. Probably everybody in this room is familiar with the spiking controls that NIST developed for QCing genomic and particularly microarray data that give you confidence in your ability to detect differences that might be small relative to the precision of your measurement. And we do quite a bit of work in quantitative flow cytometry and benchmarking materials that allow you to put, for example, the fluorescence intensity equivalence and relate that to numbers of antibodies through lyophilized, in this case, this is a lyophilized T cell, that a population of T cells that have a very similar concentration or density of cell surface receptors on it. For creating this reference material was a Herculean task and required 3 major international institutes and about, I would guess, maybe 5 or 6 publications that sort of support this analysis. So one of the things that we think about is, because reference materials can be so difficult, are there alternatives to reference materials in this field? And so, these are a couple of examples that we're working with of ways of thinking slightly orthogonally about how do we establish traceability and the ability of people in different laboratories to compare their data unambiguously and to compare data that they collect at different times. So, one problem is the use of antibody reagents for flow cytometry or imaging to identify cell surface markers that are I'm going to say putative, if I may, markers of pluripotency or differentiation state. And of course, one of the challenges here is that it's not, from my eye, it's really not clear how one evaluates data like this. So, this is one colony. What does the next colony look like? It's very hard. Does one just take an aggregate intensity measure of this, for example, or does it matter what the spatial location is? We don't have numbers for any of that right now. And even if it's just about intensity, that intensity is very sensitive to whose microscope are you using. And so one of the things that we've been doing is developing a way of benchmarking the intensity that one might see from antibody reagents to the total protein that is present in that colony. And in that way, one has a ratio that should be something comparable across different laboratories. So instead of saying, you know, yes, this is a positive, I see this, you could say, oh, my ratio with total protein for this marker is blah. And you could compare that number with somebody else's number and say, yes, that must be pretty close to an equivalent response. Another non-reference material problem that we've been working on recently is is cell counting, which is such an essential component of this field, is how many cells do you have? It's a denominator for almost every subsequent measurement that one wants to do. You know, how active is something? Well, you know, how active is it as a function of numbers of cells in the preparation, for example. But it turns out that counting cells, which seems like it's such a fundamental thing and such a relatively conceptually simple thing to do, is very difficult to have confidence in. So different methods for counting cells, different instrumentation. All right, thank you. And so at any rate, so cell counting with two different methods gives two different counts. And so which count is right? And of course, you know, it's important to know which count is right. You have to have confidence in that. And you also have to know that if you use this method or you use this method, that you have a value that's consistent and meaningful, right? And so we thought a lot about how to make reference materials for cell counts. And, you know, all the cell preparations are different. They've all got different issues associated with them. And so it became a very difficult exercise to even think about how to do this. And so instead, this approach that we're taking now is really a statistical approach that says, let's start with a model that says that if we have a sample and we dilute it by a factor of 2, we should have half as many cells present as we had from the original sample. And if we dilute it by a factor of 10, we should have a 10th the number of cells. And that linearity, which sounds kind of simplistic maybe, is a model that allows you to say, by doing multiple dilutions, I can get an idea of my precision, but if I can get a linear response function, then it tells me something about accuracy. And that way one can compare one method to another for how well they fit this model. And so it's just another way of, you know, trying to be creative about getting over some of the hurdles that are associated with this field and with the difficulty of finding, coming up with reference materials. So, you know, one thing that's fabulous about imaging, and I'm so glad that we're focusing about imaging here, because, you know, there is so much information that can come out of it, but there are also a lot of caveats. And a lot of times I find that people who don't think a lot about imaging, but use imaging a lot, don't you know, they don't use it often in a quantitative way, right? It's often used as a picture, and it provides information and insight, but it's often not used in a quantitative way. And when it is used in a quantitative way, there's still a great tendency for folks to to trust the software that comes with their microscope, or that they can download off of the web. And so one of the things that we do is well, let me just say, here, what's up here right now is an example of where trusting your software isn't necessarily the best idea. And so this is actually an experiment that came out of Wyatt Laboratories, and it showed that in response to drug, there was initially, by their analysis, there was no response that was recognized. And after they went back and improved their segmentation algorithm, they were able to see a response to the drug, which is what they had expected. And so, you know, this is an example of how if your analytical algorithm is not accurate, you're going to end up getting the wrong answer, right? And you're either going to throw away something promising or perhaps accept something that you shouldn't. And so one of the studies that we embarked on was to do a systematic study to understand how effective algorithms are, how accurate algorithms that are commonly used can be. And to do this study, we developed a systematic series of fields of view, and we used well, quite a number of fields of view. I think 71 cell objects actually went into the model, but where we imaged cells under different imaging conditions. And so you can see this imaging condition gives you really great signal to noise, and then there are various ways of degrading that image and asking how well do algorithms perform under either the best imaging conditions or not such great imaging conditions. And you know, notice that these are very simple segmentation problems. So the question was, let's apply segmentation algorithms and let's look how well they do relative to one another. Now, in order to, and this is part of the challenge that we come up with all the time, in order to compare these algorithms with one another, we have to have some sort of metrics. And it's very hard to sometimes you have to be creative to say, how do we measure how well segmentation occurred? And so this was a metric that was developed in the course of this experiment. It's a bivariate metric that gives you an indication of how much like the truth your estimate is, and allows you to then compare different answers from different fields of view with one another. To carry this out, we had two experts who did manual segmentation of all of the cells studied, and this is the result of the comparison of one expert who's up there at the 0 and another expert who's these dark dots. The light dots is the second cell line, so we did this experiment with two different cell lines, but we're just going to concentrate on the dark dots right now. But what this metric allowed us to do is that by comparing this expert with that expert, using this bivariate metric analysis, we could have a geometric value for how similar expert 2 was to expert 1 in terms of their segmentation result. It turns out that, you know, note the scale is quite small here, is that the two experts were actually very good to with one another. And we decided that if we had automated algorithms that were similar to what two human experts could do, that would be, you know, that would be a pretty good threshold value. We compared, I think, 8 different segmentation algorithms, and we could evaluate them with this bivariate metric whether the segmentation algorithms resulted in over-segmentation or under-segmentation of the objects, and very common algorithms where different imaging conditions showed lots of variability from what the experts would have said, even those algorithms that did a pretty good job still often missed finding cells that they should have been able to segment, even under the best of imaging conditions. And so, you know, this is a method that one perhaps could apply in order to make sure that you are happy with the image analysis software that you're using, and that it's going to give you what you think are the meaningful results that you're looking for. This is sort of the summary of all of this experiment, which showed that, you know, that basically thresholding did a pretty good job, but as a function of of different algorithms, that some of them were very way off and particularly bad at poorer imaging conditions. And you know, having data like this, I mean, one of the things that it affords you is that it affords you to make a decision. If it's more important to you to be consistent than it is to be accurate, and you know that you're going to have images to analyze that are not going to always be under the best procurement conditions, the best acquisition conditions, you know, you might choose something that isn't always quite accurate, but is invariable in terms of its quality over different imaging conditions. And so, you know, it's just a way of how an analysis of how your measurements are working, how that can work for you so that you get what you need in terms of confidence in the measurements that you're making. So I'm going to divert from this more general talk right now, and I want to show you 3 specific examples of imaging studies that we've been doing. So one study involves developing automated methods for characterizing stem cell colonies. And Renee, I want to thank you for bringing up the importance of automation for reducing human bias, which it can be really, really significant. The other thing that's really complicated sometimes, though, is that we have to rely on humans to provide some benchmark. And often what we find is that it leaves us with no ground truth. You know, we don't really know what's correct and what's accurate. So I'll talk about that. The other thing that I want to talk about is some of the time-lapse imaging we do, and I know that this is you know, very popular also by others in this room. And you know, one of the beauties about time-lapse microscopy is that you can actually watch individual living cells over long periods of time. And so that diversity that you can see, that variability that you can see from cell to cell, you can follow not just at one time point, but over very long times. And so there's I think, really, really powerful data that can come out of that, but they're very difficult experiments to carry out. And then the third thing I want to just touch on very briefly is a new study that we're starting that we're concerned about how to establish confidence in the imaging process that might be useful for a release assay. Okay, so so automation, you know, can reduce bias, and I think that that's it. There's going to have to be more and more automation in this whole process of cell therapy manufacturing and determining of criteria. But one of the things that's a little challenging is that often one is left with no ground truth to base their machine learning algorithms on. These are 3 images of stem cell colonies. They're embryonic stem cell colonies grown on feeder layers, actually. That's how long this study has been in the process of being completed, and this is not completed yet. So what I'm going to show you is still work in progress. In these cases, 2 experts agreed that this was a very good colony. They gave it a score of 5. This was a less good colony. They both gave it a score of 4. This was a poor colony, and they gave it a score of 2. And on a scale of, and basically the scale that they used was from 1 to 5, where 5 was best, 1 was poorest. But one of the things that was apparent is that these 2 experts, while they were incredible experts, and this was a laboratory that taught other laboratories how to distinguish good pluripotent colonies for transfer and expansion from not so good ones. The 2 experts were actually quite different when one looked at their scores in a systematic way with the same images. And in fact, they only agreed about 60% of the time. And so this was an interesting observation because, you know, machine learning is, I thought this would be an easy project. And machine learning is very powerful because you can take a training set from experts and you can develop algorithms that will allow you to then predict what an unknown sample is, how an expert would have ranked an unknown sample. But often, when we do machine learning, we often, I think the tendency is that you choose one expert to create the training set with. And, you know, we have a little joke at NIST, we're such nerds, that, you know, if you've got a watch, you know what time it is. But if you have 2 watches and they're not exactly the same, you have no idea what time it is. And that's sort of where we are here, right? Is that we know now that one expert really, you know, is not perfect, because we have two experts and they don't agree. So what does one do? With data like this, it becomes a much more complicated problem, but an important one to know about, an important one to try to resolve. And so this is basically, the scheme of how to do machine learning. I'm not going to go into any details on this, but basically you have a set of data and you have images and images with scores, and you have features that you think might be relevant to those scores that the experts gave them, that they gave these images, and you take a portion of these images and you train algorithms by doing mean squared estimates of variance. You train algorithms to choose those features that most closely match the expert score with that most closely reflects the expert score, and then you take another small set and you test it against that model. You test that subset, and then you randomly choose another subset to do training and testing, and then you choose another subset to do training and testing, and it's an iterative process until you reach a minimum variance between the testing set and the training set, and then you have a model that does some good estimate for you. We did a systematic study where, again, we looked at two experts. We looked at a couple of different segmentation methods to see what the effective segmentation method was on the models that came out. We looked at a number of features, 14 of which were specifically written at NIST to describe features that the experts told us they were using for the criteria, and then plus some heraldic features and wavelets. We looked at two different models, a linear model and a random forest, which is a decision tree model. And basically there were about 8 different sets of parameter differences that we examined all together. I'm going to show you just a subset of this. These are an example of the different segmentations. The two different segmentations would look like. These are examples of the special features that were algorithms were written to describe. One of the important elements of doing this analysis was to say, if we're going to give the experts images to look at, well, the experts took the images, actually, and then they're going to look at these images and rank them. We want to make sure that the images are of sufficient quality that there's something that the experts aren't going to be fooled by looking at them, that they're going to be a high quality image that is going to have some veracity. We're going to have some confidence in the quality of that image so that when the expert looks at it, their opinion is relevant, right? They're not looking at a poor quality image that they really can't evaluate adequately anyway. We want every image to be as good an image as it can be. And so in order to do this, Michael Halter, who will be giving a talk later today, created a material that gave us the ability to look at the phase contrast, the phase ring alignment, and give us a metric for the quality of the image. So when we have misaligned phase rings, well, I don't have an example of it here. When we have misaligned phase rings, we have not as good an image as when we have aligned phase rings. And so this was a material that was used to assure, before any data were taken in the microscope, that the phase rings were aligned and the quality of the image was good. So in this subset of models, these models involved 31 features. Just the linear model, it turned out that the more complex random forest model didn't have any advantage in this analysis. And using the automated segmentation, the images that were segmented by an automated method. And what we found was that the mean square error between the two experts, so when we compared expert 1 and expert 2, these guys had a variance of roughly 0.62. When we applied the model that was based on expert 1 scores, we could develop a model with features that provided a mean square estimate, a mean squared error of 0.42. So we could create a model that was significantly better than what the two experts could do relative to one another. And that would be an automated model that could be applied liberally on a data set. It was interesting that that only took a couple of features to have a really, really huge effect. Interestingly, is that when we used the scores from Expert 2 for the training set, that the model that we could come up with was no better, or very little, very small amount better than what the two experts could do relative to one another. And so this was kind of interesting because it says that our two experts are not equivalent to one another. You know, one expert is actually more consistent probably than the other expert. So that was a very important thing to see, that, you know, choosing the basis on which you are going to base your training set is going to be a really important decision. Now, one of the other parts of this analysis was to look at the covariance between Expert 1 and Expert 2 as they as one does the analysis of comparing mean squared errors over the course of the iteration of the analysis, that the covariance in how these two experts both respond to different features showed that that covariance suggests that there are probably that those experts are probably both seeing features and using features in their decision that is not captured by any of the features that we are using down here. And so there's a lot of information here that tells us how the models could be improved, okay, or that the models could be improved, but it gives us information about qualifying those models. Another bit of qualification that we did was to look at how robust these image features were to changes in expert or changes in segmentation method. And, you know, there were a lot of features that were not at all robust to expert or segmentation. And so this kind of analysis establishes a rigor in terms of what is the direction of improving segmentation, improving machine learning algorithms in order to have a robust response that might work over many different kinds of samples and many different kinds of laboratories, and something that's reliable. So these were some of the conclusions that we came to. You know, we now have a better feeling for what is required for a machine learning approach to be a reliable method. And, you know, certainly for something like choosing a cell therapy, it's going to be really important that these are reliable methods and that they can be applied in a reliable way to an automated process. It's really interesting about the two experts being so different because it leads us again to that conundrum of what is ground truth and how do we establish a set of ground truth that's actually going to be useful for creating automated algorithms such as we need. And you know, one question that I think of is how many experts are required to develop a training set that is reliable. How many experts should look at the same images and score them in order to converge to a training set that's really robust? More complicated questions that would have to be addressed, I think, before these methodologies are really applicable is what's an acceptable level of uncertainty in the prediction that you're going to make. And I think we would want to have a handle on that, and it's very difficult to think about how does one put an uncertainty on this kind of an operation. The other thing that's really interesting is that the features that the experts think are important for predicting the pluripotent state, you know, one can now ask, are those features correct? You know, once we can put a number on those features and we can test what those features are, we can ask, well, you know, are those the right features? And can we test those assumptions? So that has partly motivated this next study that I want to tell you about, which is using time-lapse microscopy to look at very large numbers of colonies and compare them to one another, both to compare colony to colony, but also preparation to preparation, using very large numbers of colonies for comparison, but then also to be able to examine these colonies over time. And of course, that gives us the opportunity to look at dynamic events and how the events that you might see at this time relate to events that you see in the future or in the past, and question the assumptions that we're making about image features that we think are important relative to a characteristic like pluripotency. So two of the very important questions that we wanted to ask is how similar is this colony from all the other colonies in the preparation that it's in? And then how similar is this preparation of colonies to other preparations? How representative is it and how reproducible is it? And this is something that, that takes a lot of data. So in these experiments, we have several hundred fields of view. They all get stitched together, and we're looking at a total of, in this experiment, 680 different colonies that we're comparing to one another. Michael is going to talk at great detail about this study and some of the specific aspects to it. But I wanted to just introduce this to sort of bring up some thoughts about how one can use data like this and what one can learn from it. And so, one thing, of course, is consistency is a really important thing, and what is representative and what is different. But by being able to compare in real time, I think we can ask some really interesting and thought-provoking questions that might allow us to test some of the assumptions that we make about what are good markers. So, for example, the colony analysis feature extraction study that I had just mentioned presupposes that there are features associated with the bright field or phase contrast image of a colony that can be associated with its pluripotent state, right? These colonies here are they're expressing GFP in the OCT4 gene site, and so one might think that one could take the fluorescence of these colonies and now examine fluorescent colonies that are producing GFP with the phase contrast features that are present in the features that are present in the phase contrast images of these colonies, and say, are these the right features that represent GFP expression as a measure of pluripotency? So it allows us now to go back and test some of the assumptions that we make about biomarkers. One of the other things that I think is going to be really important is to be able to do experiments like this with more than one biomarker, so that we can look on an individual cell basis over time what the concordance of different biomarkers are to one another, what's the temporal relationship of one biomarker to another as that colony and as those cells are changing. One of the things that we found with this data set, and of course when you're dealing with data this large, it's very, very challenging to handle the data and to analyze it, to visualize it. But one of the things that it has allowed us to do is to be able to identify rare events. And so by following colonies over time and reducing that data to lineage trees, one can then follow the lineage tree and identify when something is changing. That might be very difficult. Well, it is incredibly difficult to do when you've got this volume of data that you can sit at and look at for hours. You can't take it in visually, and so it has to be reduced to data that you can look at and say, oh, here's a change. let's go back now and look at the image. I can find that colony, and I can look at that image and see what was really happening in that rare event. And so Michael's going to show you some really interesting video footage of the of colonies doing things that are somewhat surprising from time to time. The last thing that I want to mention is something that we're just now getting involved in that. Well, we sort of approached this at one point in the past, but I think now we have collaborators that are really interested in some of the measurement challenges that are associated with this problem, and that is it possible to use imaging and live cell imaging to develop a release assay for a cell therapy product? One of the advantages, of course, is that dynamic information and the feature information, all of that richness. But the other advantage of using a light microscopy technique is that it's relatively indestructible. For retinal pigment epithelial tissues, which have great promise for curing macular degeneration, these are very small samples. They're about millimeter square in size. And in principle, what we've seen now from what we commonly refer to as our big data experiment, we see that we can handle very large data sets, manipulate them, and analyze them very robustly. So one could imagine being able to, in such a small sample, actually look at every single cell in the preparation. If we had robust enough metrics for what is a good preparation, what is a bad preparation, what are the things that we look at, or the features that we look at that might indicate some problem, then in principle, one could find, even if it's a very rare event, one could find those rare events in a sample prior to it being inserted into a patient. And so this could be very, very powerful. It's non-destructive. You could look at every single cell, and you could, and in principle, you could predict that this is going to be effective, or maybe it's not the best preparation. So this is going to be, these preparations are in principle going to be tested with an animal model that allows for the functionality of that implant to be determined. And that's a very important element, right? I mean, one of the things that I hope will actually come to fruition is that both what we think are very, very good preparations and what we think are not so good preparations will be tested in the animal model, so that one can say, oh, we know what the characteristics of a not so good preparation are, and we know what the characteristics of a really good preparation are, and we know something about the characteristics of those preparations that are in between great and not so great, so that they can be graded. But this is going to be, a really fundamental challenge that, but if it's successful, then, it could be I think, a really useful thing. This is actually a collaborative project that's being proposed to the borough's welcome, as one of those regulatory science things. And so, we don't even know that we'll get the funding to do it, but I think we've got a great team of people. So again, our role in that process is to assure the confidence in the imaging that will be involved in developing the algorithms that will test these samples. And so again, these tools that allow us to characterize the quality of the image are going to be essential for assuring that the image is good. One of the things that Sally Temple, our collaborator on this, was really concerned about is that once she has a preparation and it goes to a contract laboratory, how do you know that that contract laboratory is going to give you back the data that is going to be meaningful to you and that you can trust it? Well, that sounds like a lot of the questions FDA asks. And so, by having protocols that are automated, for example, to test the microscope and to provide assurance that the quality of the image is the quality that you need for doing this analysis robustly, that will provide the documentation chain and also the assurance that one can trust the data that are coming out of even a contract laboratory that you don't have direct control over. So that's the challenge and hope of that ongoing work. So, what I'd like to do now is, I don't want to conclude, I'd like to list a bunch of challenges, things that I worry about, and really stimulate a conversation about these things. I do think that there is a role for non-destructive microscopic imaging methods in this field. And, I gather everybody in this room must think so too. But there are an awful lot of challenges associated with doing this. Obviously the idea of being able to automate a lot of the process is really intriguing. But I think there's a really important thing, and it's been touched on a little bit here today already, is that I think Malcolm was the one who brought this up, is that, having rigorous methods that are going to give us confidence in understanding whether a product is manufactured appropriately or is good for clinical application is also going to improve our knowledge about biology. And, even if that were the only motivation, I think it would be a really, really good motivation. So I'm hoping that these tools will not just help manufacturing and clinical application, but will also help us understand the biology that's going on. And I think also that these tools are going to be very important for some of those really sticky things that are necessary to measure, such as potency assays. We have to have predictive models for things like that, and I think imaging is going to be a really good way of doing that, particularly live cell imaging. It's going to provide us a lot of data and give us predictive capabilities. But in order to do that, we really need robust methods, and they have to be validated to be robust. They have to really be documented as validated. And the necessary experiments that need to assure validation need to be done. And so that includes testing the algorithms, measuring, having methods for operational parameters for the instruments, studies that validate the interpretation of biomarkers is really critical. I think, there's still some uncertainty about, as Steve was saying, about really what are the appropriate biomarkers for consideration of characterization of these products. Being able to correlate the measurable characteristics that you can get out of an image and what the physiological response is of, I think is going to be a huge challenge, very difficult thing to do. But in the end, that's really what's going to make or break imaging as a useful tool. And I'd just like to stress too that, I think in all of this, there's really a great need for more research on these methodologies. And I consider that, biological discovery research, not just turning a crank in measurement science, for example. So I hope that everybody, that a lot of people could get inspired to really think about research on measurement methods as a legitimate research approach. I'd just like to acknowledge the people that have been involved in the imaging efforts. All of these folks are from our information technology laboratory at NIST. They've just been essential, incredible team that's been assembled to do all kinds of things, from stats to algorithm creation, and then our much smaller imaging team on this side. And I'd like to remind everybody, I'm going to put some flyers out, I forgot to do that we're having a NIST workshop next month that is really to go deeply into how do we look at assays and evaluate their quality. So I've spoken long enough. Thank you very much. Yes?
I just wondered if you had any discussions with people at CRH, because I'm not sure if you're aware, but I think there's some similar issues at hand. There are actually automated algorithms that are being used for readouts of histopathology slides, for example. I think a lot of the same questions and challenges were addressed in developing that technology. So you are aware of that and looking into that.
Yeah, but if you have individuals that that we could talk to, that would be great.
I can find out.
Yeah, that'd be good. Thank you. Yes.
Yeah, I think to me one of the real key issues is this, as you phrased it, correlating your assay development with physiological response.
Yeah.
You were kind of getting at that with the mouse RPE visual model. So I think a lot of it depends on the sensitivity and range of the range of sensitivity of the animal study. And I think that's one of the greatest challenges, for the kind of development of our products in general. But I also think that, like with the embryonic stem cell imaging that you were doing, the possibility of correlating with in vitro bioassays, is also a key feature. So I don't know if you're moving in that direction, or you finding partners who could look at, inward body and differentiation and quantitative assays along with the segmentation and the physical features.
Yeah. Right. No, I think all of that is prime real estate, definitely. Mm-hmm. Yeah.
I
What do you
think
think the role of
in vivo stuff is, so
Yeah.
how would you?
You mean in vivo imaging, or going from in vitro imaging to in vivo response? Yeah, I mean, I think this is why I'm so excited about this this collaboration with Sally. I hope that we can actually carry some of those ideas to fruition, because I think that it's really hard. We can do a very good job at imaging and at validating our algorithms and doing in vitro studies of biomarkers, but I think without that in vivo correlation, I think it's really challenging to know how to take that beauty and strength of imaging and really relate it to the usefulness clinically. Do you have some suggestions?
Not really, because most of them are expensive and laborious, and so. Maybe Bertha does.
No, I don't, actually. I'm just going to bring up another problem, actually. I like to thank the question about the histopathology kind of the experience, because, being an OBGYN, the classic one that we use clinically is the pap smear, the automated pap smear thing. But there were actually, they went through a giant hurdle, but you're looking at stained dead cells that aren't moving.
That's right.
So, but already with that, they had a lot of issues with, kind of quality assurance, kind of all this. But now with this imaging methodology, or this tool, if it's going to work, we still have to deal with the hurdle of the live cell and its conditions, because, I can bet that if I culture my cells and image it, and you culture the same cells in your facility, image it, they'll have completely different behaviors, different phenotype, because maybe you're using a different substrate, you're using a different concentration of the medium, temperature, all those things can alter the cell behavior, and so your institution, unfortunately, is left with standardizing all that.
No, we're not. We're not going to do that.
It's a problem.
It's really difficult. The more parameters that there are that are variable, the harder the problem gets to be, right? And so there are a couple things. One is that we're such a small organization, that what I really hope to do is see that that other laboratories that also do research and care about these things think about doing research like this, right? So that, because it's going to take a lot of data, and it's going to take a lot of really creative thinking about how do we compare your data with my data and know whether or not we're even doing the same experiment. Right? And there are ways of doing that. There are interlaboratory comparison studies. There may be intermediate benchmarks that we could discover that we could use for comparison, ways of taking the data and normalizing them to one another, perhaps through some, benchmark material in the microscope, I don't know. But it's going to take a lot of creative process, I think, to really come up with the robust methods that we can use for getting that comparative data, because if we can't compare our data, neither one of us are very sure how to interpret our data, right?
And hence the purpose of this workshop.
Anybody have any answers? Not questions, but answers?
So lunch is served, I think. So thank you.
All right, thanks.
For more, please visit us at stanford.edu.